Hi, students. I want to take a few minutes and go through with you what we did in class on Wednesday and Thursday, November 5th and 6th, 4th and 5th, November 4th and 5th. Anyway, the Wednesday and Thursday after the election. So, one of the things that we went through is the Protein Synthesis Practice 3 worksheet. And depending on what class you're in, you'll find that in slightly different places. If you are in my third period or fourth period, you will find it in the October 28th and 29th classes. And if you are in sixth period in VLA, you will find that, I think, actually in the November 5th, um, the November 5th assignment. And all of those are in person or AB live session, in person session uh, classes. So that's where you can find those. And so you'll need to open this with Cami. So choose the open with Cami, and then you'll be able to see a little thumbnail that looks like this without the drawings on it. And you'll need to open that up, and you'll need to make sure that you get all of this information into your document, because I will be checking through that to see if you did the notes as we went. So there's a whole lot of drawings on this, and I want to go over here to this the species table, and I want to show you what we're going to do with all of these pieces and parts. So the first thing that I want to look at, we're going to start out with species B here. And so what we're going to do here, you should see some blank lines here. And what we're going to do here is we're going to match up, we're going to use our DNA strand, we're going to match up our mRNA bases. So we're going to make a new mRNA strand that goes across through here. And we're going to do that by looking at 3 units at a time. And here in the mRNA strand, these are called codons. And each one of these is going to tell us what raw material to bring in. So this codon, AUG, will tell us to bring in methionine, or in this case, this will be our start codon. This will tell us that we need to start uh, with whatever product we're going to make. Once we get these amino acids together, this is what will make our protein. So this is putting together all the raw materials in order to make our finished product, which is a protein. So we're going to start with a T here. So this is TAC. And so when we start with the T, we're going to figure out what pairs up with a T. So if you have a T in DNA, that's going to pair up with an A in RNA. So we're going to type the A in right there. An A in DNA, and I have this color coded for you, the A in DNA will pair up with a U in RNA because RNA does not have thymine. T stands for thymine. So we have adenine, thymine, cytosine, and guanine. In DNA, so one thing about DNA is double-stranded, and so it has 2 strands, and there are bases that connect those 2 sides. And we looked at that in some of our notes. What we haven't looked at is RNA. RNA is single-stranded, so it only has 1 side. It's like a ladder cut in half, but it doesn't really ever have the other half. So it is made off of part of the DNA template. So here is one of our strands of DNA. So this is unzipped. And here's one of our strands of DNA. And we're going to match up the RNA bases so that we get this message that we can send from the nucleus, because this DNA exists in the nucleus. And this message has to go out into the cytoplasm of the cell and be able to take the message to the ribosomes, which function as the factory. So we have these bases that will be put in place, and that's what will make this strand of RNA. So we said that a T in DNA will match up to an A in RNA, so we'll write that in. An A in DNA will match up to a U in RNA. That's, like I said, this is one of the changes or one of the differences. This presence of the U is another difference. And then we have a C, and that's in red. And so the C will match up to a G in RNA. So we'll write the G in right there. You can see that we have done that all the way across through here. We did this in class, and I kind of worked through how this might work. So the T matches to an A. So there's a T matches to an A. Same for all three of those. The G matches to a C matches to a G, and the A matches to a U. So CGU. And then for this one, we've got G matches to a C, G matches to a C, and A matches to a U. So CCU. Now, the next thing that we're going to do is try to figure out how do we get these amino acids. So how do we read this code so that we know what amino acid shows up? And so in order to figure that out, we're going to roll up here to our codon chart. And so I've got a bunch of colored lines on here. So what in the world do all of those colored lines mean? So we're going to start off here with AUG. So A is our first codon, or excuse me, our first letter of the codon. And so here is where it says our first base. So I've got this underlined in purple. I'm drawing your attention over here to the first base with the purple. And then we're going to look at this whole entire line across through here because this is where our A is. So our A has us look all the way across through here. All of these codons start with an A. So somewhere in here is the codon that we're looking for. Our second base in our codon is a U, and I've got that underlined in blue here. So here's our U. So this is going to tell us, we know that we're confined to this purple box here that I drew in because all of those start with an A. So a U is our second letter. So we want to look at all of these right here because they all start with a U. So that's our second letter. We know then that this portion or this quarter of this whole entire box that we have outlined in purple, this quarter right here is where our target codon is going to be. Our last one here is G, and I've got that underlined in orange. This over here says this is where our third base is, so I've got that marked for you. And we know that we're looking across through here. So here's our G. So we'll follow that G all the way across, and sure enough, there we find what we're looking for, AUG. AUG. So we found our code. Now we need to know what that code means. This is like a decoder strip type of thing. So this stands for MET, which is methionine, or start. So this is going to tell our mRNA, hey, this is where the message starts. And we know that because methionine functions that way. So over here, that's how we ended up with the MET. For the lysine, how did we get that lysine? Here's our code, AAA. How do we find lysine? So if we're going to move back here, in this one, I was pointing these out with pink. So there's A for the first letter. So we know then that somewhere across through all of this is going to be our AAA. Second base is up here. So there's our next A. So that's going to tell us that somewhere within this box right here will be our AAA. Our third base is over here on the side, and I've got that marked. There's our A. Follow it across, and sure enough, there it is. There is AAA. And that codes for lysine. So LYS. And if I look over here, there it is. Okay, lysine. You can spend just a couple of minutes seeing if you can figure out how to find arginine, ARG, and proline, PRO, using this code here, CGU for arginine, and this code here, CCU for proline. Okay, now that you are back, we're going to go up here and we're going to see if we can work through this, and let's see if you guys can come up with these answers on your own. The first codon here is AUG, and that's the exact same as down here. So we know that's going to be methionine. You can pause and go through and see if you can figure that out again on your own if you would like. Okay, now we're going to try to find GCU. So in order to find GCU, I'm going to switch over to green. That's the color I've used here. So here is the G. So that tells me that somewhere all the way across through here is going to be what I'm looking for. So my second base is a C. So that tells me that this quarter of that whole set of boxes, so this quarter right here is going to be where I need to look. because I've got a G starting all these. I've got a C second, and then a U is my last one. So the third base is over here. There's my U, it's up at the top. So I go over here and follow through, and absolutely, there we go, GCU. And that gives me alanine. What I would like you to do now is to finish these two out on your own. So use GGA and find out what that codes for. What is the amino acid that GGA codes for? AGU, what is the amino acid that AGU codes for? In order to do that, you will follow through those same three steps, finding your first base, your second base, and your third base on this chart, or on the chart that you have, where you don't have quite so many uh, lines and drawings. Okay, now that you're back, the last thing that you're going to do is work on this DNA strand. And so this is just the last thing for you to kind of work through. And what you're going to do here is work backward. So if you have an A in RNA, what does that code for, or what does that pair with? What did that pair with in DNA? And that would be a T. So you're going to work backwards. So there's a T. If you have a U in RNA, there's my U. What did that code with, or what did that pair with? That was an A. So TA. Here's my G. What did that pair with? That pairs with a C in DNA. So TAC. So see if you can work backward and get those filled in. When you're finished with those, just turn this in.